Up next, we have a panel Huddle, the future of medical research and healthcare. The panelists for this session are Professor Lewis, Professor Arendt, and Sir Richard. Moderating this session will be Clinical Associate Professor Goh B. Tien from Sing Health. Prof. Goh is the Chief Executive Officer at the National Dental Centre of Singapore. Prof. Goh is also Academic Chair in the Sing Health Duke NUS Oral Health Academic Clinical Program. Let us welcome our panel and moderator. And I'll hand over to Prof. Goh.
Right, good morning, everyone. Hope you are having a good week and learning a lot from our eminent speakers. So a very warm welcome to this session on the future of medical research and healthcare. We are at a pivotal moment in history whereby the advancements in science and technology have a potential to change and reshape the way we do medical research and perform patient care. So we have with us today a very eminent panel of experts to share with us some of the future changes that they foresee in medical research that will impact healthcare and some of the challenges that we may potentially face. All right, so but before we dive into the topic itself, I would like to just very briefly introduce each of the speakers. And thereafter, they will each talk a little bit about their own personal journey in science and discovery, and particularly their road to getting their Nobel Prize. All right, so we have three Nobel laureates here with us. And we'll keep this session fairly informal, we'll keep it flexible, so you may take about 10 or 15 minutes to speak a little bit about your personal journey, or you may just say a few words, all right? And thereafter, we will dive into the panel discussion. Okay, so first up on my far right, we have Professor Louis Ignarro. He currently serves as Professor Emeritus in the Department of Molecular and Medical Pharmacology in the UCLA School of Medicine. And he was awarded the 1998 Nobel Prize in Physiology of Medicine, along with Robert Fergat and Farid Murad, for their discovery that nitric oxide acts as a signaling molecule in the cardiovascular system. He also has received the American Heart Association's Basic Research Prize and Distinguished Scientist Award for the advancement of cardiovascular science. The Russell UCLAF Prize in France for cell communication and signaling, the CIBA Award for hypertension research, the Canadian Medal of Merit, and the Golden Plate Award for outstanding contributions in cardiovascular research. He is also the founder of the Nitric Oxide Society and is a member of the National Academy of Sciences, the National Academy of Medicine, the American Academy of Arts and Sciences, and the American Philosophical Society. He also serves as a nutritional advisory board member for Herbalife International. Then sitting next to him, we have Professor Sir Richard Roberts, who currently serves as the Chief Scientific Officer at New England Biolabs, Ipswich, Massachusetts. His studies of transcription in adenovirus 2 led to the discovery of split genes and mRNA splicing in 1977, for which he received the Nobel Prize in Medicine in 1993. Since winning the Nobel Prize, Dr. Roberts has been involved in organizing a number of Nobel initiatives to correct scientific misunderstandings and promote humanitarian causes. His most recent campaign has been on the issue of GMOs, where 160 Nobel laureates have supported the use of GMO techniques to improve plant breeding practices that could greatly help the developing world. And last but not least, we have Professor Aaron Ciechanover sitting next to me. He's currently a distinguished research professor in the Faculty of Medicine at the Technion Israel Institute of Technology in Haifa, Israel. And among the numerous prizes Prof. Ciechanover received are the 2000 Albert Lasker Award, the 2004 Nobel Prize in Chemistry, which he shares with Drs. Hershko and Rose. Among many academies, Prof. Ciechanover is a member of the Israeli National Academy of Sciences and Humanities, the National Academies of Sciences and Medicine of the USA, the Pontifical Academy of Sciences at the Vatican, and the Chinese Academy of Sciences. So without further ado, I will give the time to the three professors to introduce themselves and share a little bit about their journey in science. So maybe first up, sure. Professor Louis.
Does that work? Yes. Boy, between the three of us, there are a lot of awards there. Amazing. Well, again, it's a great pleasure to be here, and I'd like to once again share with you, some of the things that have occurred in my life that have made my journey in science not only successful but very enjoyable, for sure. And I have a tendency of speaking too much, so I will try to cut it down so that I won't go past my time limit. But we can start from the very beginning. Many people have asked me, why or when did I become interested in science? And like so many other scientists I know, especially the one here to my left, I was interested in a chemistry set when I was 10 years old. I convinced my parents to buy me a chemistry set. And I used that not only to do experiments in the booklet, which I didn't understand because I was 10 years old. But you see, what I had in mind was to make firecrackers, rocket fuel, and small bombs, which I was able to do quite successfully. But first I had to entice my older neighborhoods and their older brothers to go into the local pharmacies to buy me chemicals like sulfur, charcoal, potassium permanganate, and other things, which you could easily do back then in the local pharmacy because they made practically all medications by hand instead of getting them from the pharmaceutical houses. And so because of the success I had in blowing up so many things in my home, I decided that chemistry was for me. Chemistry worked. I enjoyed it, and I kept up my interest in chemistry up until the present day. But I was also interested equally in biology. I used to drive my mother crazy because again, at the age of 10, 11, 12 years old, I would go outside. I would look for any kind of an animal that died for whatever the reason, and I would dissect it. I would cut it open, look at the inside, and see what was there. And we had, because of a neighbor, I had Gray's anatomy book, human anatomy. And I would open that up and I would compare the pictures of the organs to those in, for example, a squirrel, a cat, and I could see that they were very similar. And I couldn't imagine, how could the organs in a cat be the same as in a human? So that stimulated my interest in biology, which I continued on and on. And I guess the one thing that really turned my attention to wanting to do biomedical research was my neighbor who lived right next door. He was about 55 years old or so, and he suffered from heart trouble. And one day he came back from work and he walked to the house from the train station, which was about 2 blocks away, and he dropped right in front of the house. He had a heart attack. And that was the end of that. And so that really stuck with me, and I kept asking myself, well, how come this guy who is 55 years old gets a heart attack and passes away, and my father is 65 years old, very healthy, working as a carpenter, no sign of any problem? And so that question just stuck with me. And then when I was later in high school, you don't really learn much to answer that question, but I had in mind that, maybe in some of us, we make a molecule, a substance that protects us against cardiovascular disease, whereas in other less fortunate people, they either don't make the molecule or they make less of it. And maybe that accounts for their cardiovascular disease. And I guess about 20 years ago, we found that molecule, which was nitric oxide. Now, I'm painting a very simple path here. Believe me, it wasn't that simple. But I'm just trying to tell you, the path that I created or was created before me that led me to go in that particular direction. And so what I would say to all of you is, if you have any ideas, and you should try to pursue them. Design your education such that you could take maximal advantage of what you want to do, your ideas, and so forth. Work hard, and I think that you will be very successful in what you have to do. It's not that complicated. And I'll just leave you with one thing. The most important element, I think, when I finally did get started with my research, was the curiosity. I think that's the most important thing. You need to be curious. Always, ask questions. Why? Why did that happen? Why didn't this happen? And if something happens in your laboratory that you cannot readily explain, don't ignore it, because that kind of data are trying to tell you, what might be important. So I think I'll shut up.
Okay, well, I'm not going to tell you too much about my life, because I think many of you probably heard more than you wanted to hear yesterday, so I'll perhaps tell you about something else. But I also feel a bit of a fraud up here, because I'm surrounded by 2 doctors who, I'm not a doctor, they're real doctors, I'm just a PhD. And I could equally have won the prize in chemistry as in medicine or physiology, but the one thing that I am interested in is basic science. I love basic science. And one of the great things about basic science is that it often takes a long time before it reaches any sort of medical discoveries at all. And so we discovered splicing in 1977, and it's only about 8 or 9 years ago that the first real clinical application of the discovery came about. And that actually was made by a postdoc of mine who went and continued working on splicing. I left the field a long time ago. And he discovered that there was a problem with people who had spinal muscular atrophy, in that there was a gene that was just not being spliced properly. It was incorrectly spliced, and he figured out how to make it get spliced correctly. And as a result of that, there are a number of kids now who, well, they're in their teens, who are alive because of this discovery that he was able to correct a defect in splicing. So from 1977 until about 2015, 2016, this is how long it often takes before some of the interesting results that come from molecular biology, from basic research, make it into the clinic and make it out. And so I think for anybody who is doing basic research, you should never promise that what you're going to do is going to lead to a cure almost immediately. Just be honest about it. It may take time. You do the experiments, you do your research because you love doing research and you have fun. After we discovered splicing, I tried very hard and had a number of postdocs in the lab trying very hard to figure out how it worked, to work out the mechanism. And we worked for several years on that. We made a little bit of progress, but not much. And then we got beaten out by Tom Maniatis, who had a much better system in vitro than we did. But I switched. I started working on DNA methylases. These are the enzymes that protect bacteria against restriction enzymes. And the interesting thing is that they have two interesting properties. One is that usually they're very close in the genome to the restriction endonuclease gene, and so that means you find them fairly easily. We recently, actually, some experiments that my grandson did when he came and worked as an intern last summer, has found several of these methylases that are a long, long way away from the restriction enzyme. They're very far apart in the genome. We didn't think that that was going to be possible, but it clearly is. But the more interesting thing is that there are a lot of other methylases, and all they do is to perform some function in bacteria that we don't always know what it is. But a few years ago, we found one in Clostridium difficile, C. diff. If you go into hospital, you know that this is an infection that you may well get. It gets passed very easily. And it's a very nasty one because most C. diff strains are resistant to antibiotics. And if they're not, when the antibiotic comes along, they just start forming spores. And they wait for the antibiotic to go away, and then they start growing again. And so we found that there is a methylase in C. difficile that is controlling sporulation. And if you knock out that methylase, C. difficile cannot sporulate anymore. So this is about as close as I've ever come to doing something that may actually be medically relevant in a short period of time. There are now clinical trials underway from a couple of companies that have worked out a potential inhibitor. We don't know whether it's going to work or not, but that's about the only really thing I would say to you that qualifies me perhaps to sit up here and talk about medicine. So thank you, and I'll pass it over to you, Aaron.
So on the background of a typical Jewish mother, my mother wanted her two kids to be either lawyers or physicians. So that's traditional. So my eldest brother became a lawyer, and I couldn't disappoint her. So I went to study medicine. And actually, it was quite interesting. And following, after I completed my studies, I went to become in Israel, you know, everybody has to serve in the army. So I was, for several years, a military physician, a combat physician. And then I decided to go to the most extreme side of medicine, which is surgery. So I was trained as a surgeon. And then I realized that there is a problem in medicine. And most of the patients with real diseases, I'm not meaning when you are involved in an accident and you break your leg, are coming too late. When you see them, it's too late. If you take, you know, Lou was talking about myocardial infarction. When a patient succumbed to a myocardial infarction, the disease started decades before. He started already to accumulate cholesterol in the coronary arteries, and the last event, the final occlusion, is the event that was shown up symptomatically, but the disease started much before. The same for Alzheimer's disease, for neurodegeneration, the same for cancer, that the first mutation occurs in many diseases, in many malignancies. And I had this naive thinking that I want to understand why diseases start. And the answer, I thought, should be in the lab. I must admit that it was either a naive or a courageous decision to depart medicine at such a late stage and to go to the lab, because in the lab, you know, you are not studying how to cure cancer, and you're not studying big questions. You are concentrating on some nitty-gritty corner of nothing. So I went to train with a very young person, with Avram Hershko, and he was interested in protein degradation, when the whole world was concentrated in protein synthesis, on the central dogma of biology. Israel at that time, in the 70s, was a small country, relatively poor, so we tried to stay away from the highway. But we asked a very specific question, and that's not how protein are degraded like in the stomach, because in the stomach they are degraded indiscriminately. You know, you can pour into your stomach, you can go to a 3 Michelin Forks restaurant and pour, you know, $200 worth of a nice wine, accompanied by a $200 worth of a nice steak, and it will pass through your taste buds for 2 seconds, and then it will all be gone. So we were not interested in that. We were interested in specific degradation. how the cell destroys protein in a specific manner, namely mutated protein, misfolded protein, protein that are faulty, and also protein that are healthy but are not needed anymore, like cell cycle drivers, transcription factor that completed the function and need to be removed. Then, in order to cut a long story short, I will not go into details, but we discovered the ubiquitin system that little by little grew into a major biological platform, and many drugs are currently in the market to treat mostly malignancies, blood malignancies, but the brain is already coming behind the corner. So I must admit that my dream at the end, my naive dream and my risky one came true, but not by us. So we didn't discover the drug. Now we are developing drugs in my lab and so on. Other people took over what we discovered and went on. So initially they discovered drugs, diseases that are related to aberrations in the system, and then the pharma company took over and started to discover drugs. That's what always happen. And most of the drugs are aimed to remove faulty proteins. Let it be in cancer, it are oncoproteins. You know, most cancers are driven by oncoproteins. In the brain, it will be aggregated proteins. You know that all neurodegenerative diseases have one common hallmark, which are aggregated protein. Let it be in Alzheimer, Parkinson, Huntington, ALS, and so on. The difference between the diseases is the type of the protein and the site of their accumulation. So all of a sudden, the destructive system that nobody was caring much about became kind of a mainstream, not only in biology, but also in the pharma. So as a physician, for me, it's nice to see it, because as a surgeon, you know, you operate patient one by one by one by one, and it depends on the type of operation. You can stay in the OR for 12 hours if it's a big tumor, or 3-4 times a day, but each time is a patient. And if you are lucky enough, then you shouldn't forget the element of luck, beside passion and perseverance and being with the good people at the good places. Then you can see that your studies, like Lou told you wonderfully yesterday, can affect quality of life and lives of millions.
Thank you very much for the very interesting and insightful sharing. One question I have for you is, what do you see as the greatest unmet need in medicine and healthcare today, and how can research potentially fill this unmet need? Anyone can.
Well, I've thought about this, and I'm not really a practicing physician anymore, but I think one of the greatest unmet needs is I think our interest should be more in preventive medicine. I want to take a different approach. If you can prevent a disease from occurring, that's a lot easier than treating it, reversing it, curing it. But how do you prevent disease? Well, to prevent disease, you have to have a really fundamental understanding of the pathophysiology of the disease. And yes, researchers pay attention to that, but I think that groups of people, individual people, should pay far more attention to preventive medicine. And I think in that way you could save, millions, tens of millions, hundreds of millions of lives. And I'm going to give you one example. Only one, because it's the only example I know. And that's cardiovascular disease. Are you aware that 90% of cardiovascular disease is preventable? 90% of cardiovascular disease is attributed to poor lifestyle. Not eating healthy, not eating the right foods, eating the wrong foods, and on top of that, sitting down like we're sitting down 7 days a week. Now, we do that because we're at a conference, but we need to be active. Humans were meant to be active. Humans were meant to exercise You just look back on evolution and you can see that, and I'm not going to give you another lecture on evolution. But all I want to say is that we really should focus more on preventative medicine. If the people of the world somehow would eat better, would exercise more, I mean, we could cut back on tens of millions of deaths, unnecessary deaths, very costly deaths due to cardiovascular disease. So I'm going to stop there.
So I would add on what Lou said, and prevention also include early detection. And there is a whole world now of biomarkers that we can detect like malignancies in circulating DNA or circulating tumor cells. Imaging, for example, there is a big discussion about imaging, for example, patients who are smoking, people who are smoking, just to detect early lung cancer. And it's true that lifestyle is fundamentally important for prevention, but then what about genetic diseases? And I think that this is another issue that is being added. But remember that CRISPR is behind the corner. We should use it very carefully. Probably along the discussion, we shall get also in ethics. In medicine, ethics is a very heavy issue. You know, changing the genome and so on, to whom, by whom, what is the red lines that we shouldn't cross ethically, and so on and so forth. But diseases that can be discovered that will erupt decades from now can be prevented by maybe by editing genes. So this is another aspect. So this is just to add on preventive medicine, which is much broader than, of course, just exercising, keeping the right diet. With all that, I want to get back into ethics and to talk about accessibility, just in one word. You know, science is a beautiful profession. We are sitting in labs at Harvard Medical School, in very sophisticated, in the Max Planck, very sophisticated institutions. But we shouldn't forget that 4 billion people, half of the world population, do not have access to clean water. So, you know, whatever we achieve in the lab is not necessarily getting to all the people. So we have to take care also of this issue, and even right diets and lifestyle and so on. It's a matter of education, resources, cooperation between countries. And then the real challenge, or one of the real challenges that are on the desk, is of course brain diseases. I believe that cancer, vascular diseases will be defeated. Brain diseases that are kind of aging or more advanced age diseases are still on the desk, and we don't know much about them, and they are really a major problem of this debilitated old population that managed to use the advantage, the technological advantage of medicine, reach the age, and then they encounter this wall of neurodegeneration.
Okay, well, I'm going to take a slightly different tack. I'm 100% behind diagnosis. I like that. I think one of the ways in which we very soon will be able to do a much better job of diagnosis of genetic diseases is because we will understand much more about the human genome sequence. We will be able to sequence everybody's genome when they're born. If they have a genetic defect, we will know about it, and so we can think of ways to defeat it, and then at the same time there will be research opportunities to figure out how to do that. But I'm actually in love with bacteria. I love bacteria. And you have to realize that none of you sitting in this room today would be alive if we killed all the bacteria that live with you. You need these bacteria. I love them. They have a bad name, and the bad name came because when microbiologists first started looking at bacteria, all they did was to link them to disease, find out anthrax, all sorts of bad things. And so we ended up in this sort of feeling that bacteria were bad. But in fact, 99.99% of all the bacteria living in your body and on your body and in your organs and everywhere, they're good. One of the interesting things, we know that there are bacteria that fight cancer cells. You have to remember, we are the home for the human microbiome. They don't want us to die. They want us to live forever so that they have a home. And there are bacteria that have been discovered that will fight cancer cells. Would be nice to find out more about that. We also know that the brain has more connections to the stomach and the intestinal tract than any other organ in our body. And in those tracts, there are bacteria living, and they're talking to our brains. I want to know what they're saying. I want to know what they say to one another. I want to know how they communicate, and I especially want to know how they speak to our brains, because this may also be a significant part of many brain diseases that come along. So I'm looking at it from a very basic standpoint. I want to know how these things work. And I think if we know how they work, we're going to make some very interesting discoveries that will have lots of medical applications, and maybe it'll take 20, 30, 40 years before we have these applications. But just imagine if you could be fed a bacterium that would make a big change in what was going on. And I'll give you a good example of that. C. difficile, which I mentioned earlier, is an awful disease. If you get a chronic infection with C. difficile and you can't get rid of it, you have constant, uncontrollable diarrhea. And so people who get it go home, they stay home, they never leave their house because they don't know when they next have to go to the toilet. It turns out that you can solve that by using transplants. You can use fecal transplants to actually kill these C. difficile. We'd really like to know how that works. We don't know how that works very well. You've got quite a lot of bacteria living with you, literally thousands of bacteria, different strains living with you. There are a few predominant ones, but lots of small ones. When we can find out more about how these things work, how they help us, how they're going to allow us to grow, I think we're going to have some great success. And one of the nice things about bacteria, they're small, they have small genomes, they're actually easy to work with. You can work with them in the lab, a lot easier than trying to persuade Aaron that we should be doing some experiments on him. I'll stop there.
Okay, thank you very much. So on that note, perhaps we would like to hear your thoughts on precision medicine, which is garnering a lot of attention currently. So what do you think would be the role of precision medicine, which is to tailor treatment based on a person's genetic profile, or maybe even the microbiome, or even their lifestyle, to assess their risks and to tailor, say, a preventive strategy or treatment based on their profiles? Do you think that will become the mainstream of treatment moving forward?
I'm going to defer to the two gentlemen here because I really don't have any expertise in the area, but just want to point out that I think those approaches are extremely necessary moving forward.
Well,
I think that, you want to take over? So precision medicine is a kind of a buzzword, but it's yet just another development in medicine. That's it. And it was enabled by initially understanding or exposing the human genome, and then what came following it, the transcriptome, the proteome, the metabolome, and the metabolome is the lipidome and the glycome, and so on. So we can profile patients now. Initially, I remember as a surgeon, you had a tumor in the breast, so you take the breast away. A very mutilating procedure and traumatic one. And now we start to realize that, again, by technology, that there is breast cancer A and B and C and D and E, and one is related to mutation in the estrogen receptor and can be treated with tamoxifen. Another one is related to mutation in the EGF receptor and can be treated with Herceptin, which is an antibody that will cluster the mutated receptor and kick it into the cell to be degraded by the lysosome and the ubiquitin system. So now we can profile patients according to the disease that they have, but not the disease, but the mechanism that underlies the disease, the molecular mechanism. That's the whole story about precision medicine. So we can be much more precise, avoid mutilating procedures, and also treating the patients with biologics, which the treatment itself is less traumatic. These are not chemotherapeutic agents. The side effects are much lighter, and so on and so forth. So it's a magic word. We are already into it. It's not that one day the New York Times of January 1st, 2027 will say, yesterday evening we started the revolution in medicine. No, we are already in, and as we understand more and more about diseases and their mechanisms, we are developing more and more drugs. And there are many other technologies that are coming to help us, because there are some mutations that are not druggable yet. For example, KRAS, one of the most awful oncogenes, is hardly druggable, but now other technologies came to our help, like AlphaFold. Now we can crack the structure of a protein with the hit of a keyboard. This was the Nobel Prize in chemistry a few months ago. So if you know now the structure of the protein beforehand, it took years. Think how many years Ada Yonat spent cracking the structure of the ribosome. Now you can do it, again, based on wet information that was accumulated on the structure of numerous proteins in the lab. And then if you have the structure, you can now design by computational chemistry a drug to modulate it. So the way to new drugs is much easier, which also will pave the road to a much shorter and cheaper way to develop new drugs from mutations that we still cannot treat, or we still don't know much about them. So you see, it's just a technology, but the technology, again, I must come back to ethics. We are in science, indeed, and we are in the lab, but behind the lab in medicine, stand patients, and I will not go into it. We may have time to allude on it later, and patients are human beings that are stricken by disease, and their world flips upside down. And we shouldn't forget it for a minute. And personal medicine has several issues with it. It's not like a rosy road. One issue is, of course, pricing, because we have to, breast cancer is not one disease now, but it's several diseases, and it requires development of several drugs that have to replace the scalpel of the surgeon that is a cheap instrument. And each development will still cost money. Each drug will still cost money. This is one issue, and it has an ethical issue, and that's what do we want to know about ourselves. You can walk into the emergency room, and in the next test that you are going to undergo, whether you want it or not, will be kind of either SNP analysis or partial sequencing or that kind of sequencing, and the doctor will come to you. I always bring kind of an example that I sit in my lab and I have a chest pain, and I think maybe it's a myocardial infarction, so since our hospital is just next to us, I walk to the ER and ask the doctor to test me, and he takes an ECG and takes some canonical enzyme, and he say, Aaron, I have good news for you and bad news. The good news that you don't have a heart attack, but we found an APOE4 gene, a double APOE4 gene that can predict with very high sensitivity that you'll get Alzheimer in 10 years. So then, Jesus, what do? I came with a chest pain and I exit the emergency room. So the issues are here very tricky. Whether he should not tell me because there is nothing to do now about Alzheimer. So to whom belongs the information legally? And if it will stay in the computer of the hospital, somebody may hack into it and will call me tomorrow and say, Aaron, if you don't pay me ransom, I will put this information on the network. So think about it. And even if he tells me that, then what do I do with it? I go to my employer, I go to my health insurance, I go to my wife, I go to my kids that may know about it or so. So you see that there are many issues here. I'm not going to delve into it. There are issues that are beyond the lab bench, but they nevertheless belong to the core of the problem. There is a gene that is mutated, and the same for BRCA1 and BRCA2, BRCA1 and BRCA2, and so on and so forth. And the repertoire will grow exponentially as we are exposing more and more diseases. So you see that many issues have to be solved, but I think that there is old Jewish say that tells us that the more knowledgeable you are, the more troubled you are. So, you are not here to frighten you, of course, but in order to answer a question in medicine, we first have to ask the question. So now we are in the phase of asking the question, and medicine and science will find solutions to all that, but they are not going to be simple. The more we are advancing, the more complicated it becomes.
Okay, well, that just goes, sometimes it's better not to know. Sometimes you don't want to know about some of these diagnoses that come along. But I would go back to sort of the very basics. if we're ever going to really use the information in the human genome sequence to make predictions and to solve problems, figure out how we can diagnose things early, we have to understand what are the genes that are present in the human genome. And although there's a relatively small number, according to some estimates, we're now discovering that there are actually many more small genes, small proteins that are made, we don't know what they do. Small RNAs that are made, we don't know what they do. And now is the time when we really can focus on doing some research on specific regions of the human genome, figure out what the genes are doing, what's going on. It's not trivial. The human genome is pretty big. E. coli, which is the best studied bacterium out there, only about a little over half the genes can we say we know what they do, that we can really be confident about what they do. I would like to think that before I die, and I plan on living for a long time, I might say, although I don't do any exercise, so sorry about that. Although I do try to walk quickly from one terminal to another in the airports, but that's about it. But I'm hoping that we will actually understand how a bacterium works before I die, because I think knowledge like that will actually be very helpful in understanding how other organisms, including humans, work. And that is something that is, I think, doable. It really is something, if we focus on it and concentrate on it, we really should be able to understand. And when I say we understand how it works, I mean we can write a computer program that will absolutely respond in the way that an E. coli cell will respond to whatever stimulus, whatever we give it to eat or whatever. And so there's a great deal of basic research there, which is what I love doing. There is a great deal that can be done that will have significance in the medical field later on, whether it's on bacteria, whether it's on humans, whether it's on pigs, on rabbits, whatever. There's going to be a lot of stuff that we can learn that will help us with medicine in the future. So I want to go back to basics. I want to see some of you guys doing some of this basic research, because I guarantee you're going to make discoveries. You will make interesting discoveries.
Thank you. Yeah, so I think Prof. Aaron did mention a few times about ethics in research. So probably we want to touch on that topic. So as we push the boundaries of medical research, right, more and more, we talk about us now playing God, right, to do gene editing and things like that. So how big do you think that problem is, and what framework or policies should be set in place to ensure responsible research among the community?
Again, I mean, taking those approaches are essential. I think we all believe that if we're going to be able to look ahead and solve problems, prevent the occurrence of disease, find unique ways to treat disease, prevent genetic diseases and so on. And of course you're going to have an ethical problem because there's so many human beings who are not honest. And if we were all honest, we wouldn't have a problem. We could all be working on that and make such incredible discoveries. But the ethics of it is a serious problem. And I just don't know how we could set up, how we could establish ethical principles such that we could continue to do this research in a straightforward manner that wouldn't lead to a problem. All I can emphasize is that it is essential to continue this research. I mean, just imagine developing procedures that would abolish all genetic diseases. I mean, just think about that. We can do that. Yes, there will be ethical concerns. Should we allow the ethical concerns to slow down our research in developing these therapeutic strategies? Science is amazing and will always move forward. You cannot stop science. Never. It'll always move forward at a rapid pace. And we as human beings just need to monitor that closely, monitor it very closely, and do the best we can to prevent the abnormal use of this kind of technology. But again, I do not know how we could approach establishing ethical principles other than governmental regulations. You can't do this, and you could do this, but you can't do that. I just find that government regulations don't work. But again, I don't know how we can solve the ethical problem.
You raised it, so
I'm to be blamed for raising the issue of ethics. I want to pick up on where Lou left it, and that's whether we should stop or limit science because of ethics. And I completely agree that the answer is no. But there is a but there. And the but is that science should be self-limiting, and mostly in democratic countries. I think that democracy is not an ideal system, but it's an optimal system of checks and balances. You are living in a free society, you have students, you have funding agencies, and I think that this community altogether should make a decision what should be done and what shouldn't be done, like cloning people and so on and so forth. I don't want to go to the extreme, but I think that free society that has a real parliament, not autocratic regimes, will find, and the court and the Supreme Court and the three authorities that are separated from one another will have the community power to decide where are the red lines. So I can bring one example. There was an example in China of somebody that did a CRISPR-Cas on two young embryos, very young embryos of Lulu and Nana, these are the code name, and it raised a huge anger because it's much too early. And in order to protect them from AIDS, I think, during passage through the birth canal. And medicine knew very well how to prevent it anyway, and it turns out, actually he wanted to remove the entry receptor of the virus into the cell already in the embryo, so when the baby is being born through the birth canal of the mother that is infected, shouldn't catch the infection. So first, medicine knew all about it. Second, there are two copies. So he managed to do only one and the other baby not, and it was raised huge anger, and then the CRISPR people themselves decided what should be the red lines, like Jennifer Doudna and Emmanuel Charpentier. So scientists themselves decide, living in democracies, what is the red line, what should be allowed and what shouldn't be allowed, and what is allowed and what people are trying today are, of course, genetically inherited diseases that are fatal or carry very harmful defects in the embryo. Let's talk about, thalassemia or sickle cell anemia or SMA that Rich mentioned, that is now can be alleviated, and so on and so forth. So we do need control, not stopping science by any means, but really, decide together what is appropriate and what is not appropriated ethically, because we don't want to cross these lines.
Maybe I'll tell you a little story about this. So back in 1975, when recombinant DNA was first being discovered and used, there was a lot of concern, and I was one of the people who were quite concerned about this, that all of a sudden it seemed it would be possible, for instance, to make very deadly viruses in the lab, accidentally, maybe even deliberately if you were a terrorist. And there was a lot of concern about this. We had a conference at Asilomar in which we got a large number of people, large number of scientists to get together. We had journalists, we had people who were professional ethicists came. And one of the outcomes of that conference was that there were going to be 4 biosafety levels. Biosafety level 1 was essentially you could do what you like in the lab. Biosafety level 4, you had to go into a special facility and really take great care. And there was a lot of amusement at that conference because Sidney Brenner, who you all know very well here in Singapore, he was at the conference and he had written down some prescription for biosafety level 5 and biosafety level 6 on a sheet of paper, and he was handing it, was going from one person to another, and there was this ring of laughter going around Asilomar. For him, biosafety level 5 was where you went into the facility, did your experiments, and then you autoclave the investigator when he comes out. Biosafety level 6 was where you autoclave the investigator before he went in. So it is an important issue. I feel strongly that we should be very careful. CRISPR is very good, but it's limited. It's not yet at a point where we can say for sure that we're going to put in a gene, knock out a gene, absolutely without any problems. It will get better. Scientists will get better at doing this sort of thing. And so all of that is very good. So we have to be quite careful. And as Aaron says, if you have a genetic disease that we could cure by maybe making a change and stopping sickle cell disease or stopping something else, we should. But money is a huge problem. And I think one of the things at the moment that concerns me from an ethical standpoint, actually, is that the pharmaceutical industry, actually, they're not interested in you getting better. They don't want to cure you. What they prefer is to have a drug that you will take for the rest of your life to provide them with profits. And this is why they've done very little research on antibiotics. Antibiotics work, and it's a big problem. And I think I find the ethics here and the fact that the pharmaceutical companies and many of the people who will be doing some of these CRISPR things, they're more interested in money than they are in making sure that we're all well. And I think that's something we have to deal with. We should not just let them get away with that.
Thank you. So on that note, I think we shall leave the rest of the time to the people in the audience. Yeah. Oh, okay. Maybe.
Hi, good morning, speakers. Very thankful to be hearing about your inspiring life. I just wanted to ask this question about how do we go about bringing change when it comes to biomedical research, diagnostics, and all that, because very often a lot of things that people come up with are just become another paper, and it just goes about not really applied. And how do we convince people to change from traditional methods that people still stick to, although they know that it has many problems and issues? Thank you.
Okay, so as I understood it, what you were saying is that mainly people who are doing research in these areas are more interested in just publishing the results and are not interested in making sure that those results get translated into better medicine. Is that what you're saying? Yeah. And, ultimately, it's up to us to some extent. We do a basic research, we make a discovery, we find someone to convince that they have to take it further. Most of us who do basic research do not do the follow-up on the clinical side of things, but it is possible. Some people, like to do that and then like to follow through. But it would be better if there were some really well-established channels by which basic research could be conducted and then the results moved into the clinic. And in principle, this is what industry is supposed to be doing. This is what pharmaceutical companies and others are supposed to be doing. But they don't always want to follow through on what we think is good. So you have to be a bit of an activist. You've got to find some people who you can talk to and who will then take this idea and convert it into practical medicine.
I think that the main drive for research should be curiosity and the pursuit of knowledge and not application. And application will come if it's proper. So for example, when we studied the ubiquitin system, we didn't dream of a disease, we didn't dream of a drug, and actually we didn't develop the drug and we didn't discover the diseases. Other people took over, and that's the nature of science. I think that in the word applied or translational, the nature of this research is already embedded. What is applied? That you apply basic into something practical. What is translational? That you translate a language to another language, right? So you translate the basic findings into something that is practical, that people can use. So I think that we should erase these two terms from our vocabulary and use good research or bad research. And good research, some of it will be translated, and some of the research, will never be translated, but it adds to our understanding, whether the universe is expanding, are we going to develop drugs out of it? Not. But it's extremely important.
I don't know. Brian is going to do that.
Yeah, but it's extremely important to understand it. So, we want to understand the universe. So some people are doing translational, some are doing basic, their research is being translated. So let just people fly on the wings of their imagination.
Right. I mean, I can add to that. When you do your research and you publish it, don't worry about where that research is going to go and who you can communicate with to be sure that this concept can be tested here, there, or whatever. Just do your work, publish it in a good journal, and believe me, if others will recognize the importance of your work, and then will follow through. And then the first thing they will do is try to reproduce your work, and I'm sure that they will be able to do that. And then this will go further and further, and eventually something good will have happened with the research that you did in the first place. And you'll still get credit for doing that. I mean, I remember when I was a urologist at UCLA came over to me to ask me, what is the neurotransmitter? What is the chemical released from the nerves that stimulate erectile function? And the first thing I said to him is, I said, why are you asking me? I'm not a urologist, I'm just a pharmacologist. And he said, well, the neurotransmitter is not known. And I said, all right, well, why don't you go figure out what it is? And so he said he didn't know how. He asked me some questions, and then I went back to the lab, and I'm thinking, I might have an idea. And so we did some experiments, and sure enough, we found, as I explained yesterday, if you were there, that it was nitric oxide. But at the time, I didn't even think about developing a drug that would increase nitric oxide and therefore stimulate erectile function. But the word got out, and I have to give some credit to some pharmaceutical companies. One picked up on that, and a few years later, we had Viagra. So I'll take the credit for that. That can happen.
Yeah, I would actually take a slightly different point. I think what you were asking was if you were doing something, make a discovery, and you see an application, right? You see it. How do you make sure that it goes further? And I think what I said before, you have to push it. You really have to push it. When I was a young researcher and we were first getting DNA sequences and RNA sequences, we needed some computer programs to put those sequences together and figure out how something that was maybe a kilobase long could be made from something that was 100 nucleotides long. And I wrote some programs, but nothing really special, and so I encouraged others to write better programs, people who were professional computer programmers, to go on and then to put together, and now we have excellent software for doing all of that. And that is something that you can stimulate, you can get it going, but you don't necessarily have to do it yourself. And I think that's kind of the thing you were talking about, yes? Okay.
Thank you, professors. Thank you for sharing. I'm asking some questions about the healthcare because what I'm doing is about software wearable electronics. And we see this as a fascinating future for the human use healthcare monitoring, like the vitals, like ECG, right? So we think that it can improve the convenience for people at home or for patients. But after talking to some doctors, I think some of them are very optimistic, but still there are others have some concerns, right? So they have several things. First is they are questioning about the reliability of this wearable devices data because they are always golden standards. So they don't kind of believe this data we collect from the devices developed. And second is I thought that doctors prefer the face-to-face with their patients because they can actually touch them and know exactly what they're suffering and what they want. So they feel that if you have these home-use healthcare devices, they may not have this kind of direct diagnosis. And third way is that because what they face with the home-use devices is all this data, and data is quite cold. And sometimes they can improve the efficiency, but they also think this might increase their workload in the future. So these are their concerns. So my question is, what do you think of the future of these wearable devices for home use healthcare? And do you have any suggestions that we can convince this doctor and to also believe in the future of these home use devices like us? Yeah, thank you.
Okay, so let me take a stab at that. You'll notice I'm not wearing an Apple Watch, and my wife wears one. I will never wear an Apple Watch. Personally, I prefer not to know.
Well, there is a long line, so I'll be very short. The world is in a great shortage of physicians all over. In my country, I know the numbers, and we are trying to overcome it. We are trying to overcome it by educate physician's assistant. We are trying to overcome it by remote medicine, by having home devices, and so on. It's true that it's less humane. I mean, especially sick patients, they need, tapping on their shoulder. They need to see face of other human being, and so on and so forth. And I think that we are choosing the best way in between. There is much into home devices because you can follow yourself. You can also educate people to be more aware, to monitor their blood pressure. The blood pressure, the high blood pressure is known as the silent killer. People don't know that they have it, and it can kill them, either from heart attack or from a stroke or from whatever. So unlike my good friend Rich, I think that I would like to know, maybe not everything, but some fundamental things, yes, I do want to know. If people are educated to take good care of themselves and even diet and so on, they can weigh themselves, report their weight to the doctor. In remote, the work of the doctor is much more efficient. He can see many more patients per time unit. So this is one way to overcome the shortage, but you are right. I think that in severe cases, and I always remember, we shouldn't forget, and I'm sorry for being, for dragging it a little bit. I always remember Saturday night when I was an active clinician. What was Saturday night? I was sitting in my home and going over files of patients that I got the results already before, and I would go to see them on Sunday. So that's what it. So they were sitting in their home, talking to the families, looking at the TV, and I'm looking at the file ready for the clinic of next morning. And what I had in front of me are, from time to time, fatal or very bad diagnosis. Let it be Alzheimer, when I was sitting with a neurologist, but as a surgeon, pancreatic cancer, hepatic cancer, ovarian cancer, when I was sitting with a gynecologist and so on. And I knew that the next day in the morning, when I'm going to meet these patients, the world is going to flip upside down. This cannot be done by remote. No way it can be done by remote. You need to sit with appropriate courage with the family of the patient and talk to them and tell them the truth. And because they are human beings that, as I said, their life is going to turn upside down once they are hearing the little sentence that they have pancreatic cancer. And if they don't understand it, it's the doctor that has to explain them that this is a fatal disease. And the course to death is going to be rather torturous. So we need still, as I say, we are coming back to ethics because medicine at the end of the day is not a lab bench. Medicine is treating people. And we need to be very considerate. But for, if you have a flu, you don't have to run to the ER. And there are better and better monitoring ways in order to diagnose it, to recommend. You can write prescription, you can send them via email, they can go to the pharmacy and get the drugs. Medicine, remote medicine, is good for most of the cases because luckily not all patients have pancreatic cancer. So at the end of the day, we have to find the midway.
Thank you.
Good afternoon, panelists. I'm from Japan, but I'm Bangladeshi, and I was literally on the tip of my seat when you guys were talking about different topics such as the ubiquitin, the endocycle. I am a 40 undergraduate student dealing with immunology and pharmacology, and as Sir Richard Roberts mentioned about the gut microbiota, actually my lab has actually find out that insoluble fibers, we are advised that we should take fibers for a gut microbiota, but we actually found that insoluble fibers contribute to anxiety due to the via the brain and the gut axis. So it really shocked me that how like it's controlling or contributing to mental diseases and so on. So it's really interesting to see here. And one thing that I wanted to know from all the panelists over here is what one saying or opinion do you feel strongly negative in the field of medical research and healthcare? Something that you have really strong opinions about, or something you wish like they would stop saying it, or you think is completely wrong?
I'm sorry, I didn't get the question.
Yeah.
It's amazing that most questions are related to medicine, and at least none of us is a practicing doctor. So maybe we should bring one to the panel.
But at least you used to be.
Well, at least I studied it half a century ago.
So if I understood what you were trying to say is, what should we be prioritizing in terms of the research that we're doing? Where should we be looking in order to really make some big advances? Does that sort of sum up what you were asking?
I'm just wondering about your personal opinion that you strongly feel negative about rather than like positive, like maybe a saying or any advice or anything like that.
Okay.
Go ahead. I could answer all of this and go on forever, but you guys should speak too.
I think Aaron has so many great opinions, I think he should answer this.
I think all Nobel laureates are very positive people. They don't have any
That's
negative.
probably the answer, right. Sometimes it's difficult, to understand the question. So when you're asking the questions, try to use as few words as possible, okay? And just make your point succinctly and then ask a key question so that we could sort of understand it and therefore give you a much, much better answer. So I apologize if I don't really understand, what you're asking.
Yeah. Good if we took that same advice in answering the question.
Maybe Brian can pick it up.
Good afternoon,
Okay, so maybe we'll
professors.
move on to the next, yeah.
Thank you for your sharing today. By way of introduction, I'm studying social and cognitive neuroscience, and it is a very young field of research, particularly in the area of well-being. So to this end, most of the papers that I read today will quote small sample size as one of the limitations of research. And when you have most of your paper quoting the same limitation, it is difficult to find a way forward. So my question is, for us young scientists who are going into very young fields of medical and healthcare research, what is one advice you would give when faced with confusion or not knowing which direction to go to due to how new the field is? Thank you.
Well, I think I can start off by saying it's, you have to start off somewhere, and you're going to have to make up your mind as to what field you want to get into, and then you begin to work in that area. And once you start to work in that area, I think it'll be self-evident which direction you may want to go in. I mean, we cannot tell you what area would be better than others. There are areas of medicine, I think, that need a lot of work that one could go into. There are other areas where there's been so much progress already. Maybe you don't want to pick that area, you can pick some other thing, but it's really up to you to understand the field, do your literature or research or searches, and then try to make some decision as to, which area you want to explore, if I understood your question correctly.
I think I have a simpler answer to this question, which is a very important question, and that it doesn't matter. It really doesn't matter. I think that what really matters is to train with good people in a good place. And then what you are gaining during, especially the graduate studies and then the postdoctoral fellowship, are tools, mostly thinking tools and problem-solving tools. And then you can take them wherever you like. So, for time to time you can go to the absurd, why the banana is curved. Yeah, it's very interesting why the banana is curved, because it may lead you not only to solve the problem, to do genetics, maybe there are some physical forces and so on, but maybe then to go to diseases of kyphoscoliosis, where patients have this curved spine. I don't know. But I think that being with good people, of course you need luck. At very good places where not only the vertical environment is good, so your mentor and the PIs are good, but also the horizontal environment is good. Many good postdocs to discuss, lots of noise in the lounges and lots of discussion. That's the best, and then it doesn't matter. Heart diseases are important, expansion of the universe is important, chemistry is important, they're all important. So I really, of course you should follow your gut feeling. You have to know, what you are good at, and that it should really drip from the tips of your fingers. But what specific subject, in my opinion, it doesn't matter. I did my postdoc, I was lucky to have a very good PhD mentor. And then when I had to go to a postdoc, I decided exactly what I'm telling you. I went to MIT, to a very good mentor. And it was like, wow, it was a temple, the mecca of science. Not only in the lab. In my lab, there was another guy who also won the Nobel Prize, Jim Rothman. So you see, from one lab came 6 members of the National Academy of Sciences, 2 Nobel laureates. The environment was amazing environment. You could get consultation, probes, reagents, just in a spitting distance. And that was enough. And then it doesn't matter what you do. So I think that the secret, again, I repeat it again, good mentors and good environment. And typically, good mentors are in good places.
Thank you.
Hi, my name is Daniel. I'm a gastroenterologist, so I practice medicine. I hear a lot that the disease viewpoint that you have is very mechanistic and it's very upstream. But the reality of medicine is that it's very organ-based. And so, like, say, a gastroenterologist like me see fatty liver, and my friend down the corridor see heart disease, and another one see diabetes, but probably it's about the same disease. How do you think we can organize medicine to make it more mechanistic rather than organ system based?
I think he's asking how we could organize medicine better than the way we're doing it at the moment. Is that about right?
Yes, that's
Yeah.
right.
So organizing medicine?
Yeah, organizing how we solve problems, how we look at particular problems.
So you're thinking taking a more mechanistic approach rather than organ system approach? Well, I think that both are important, but again, I'm not sure precisely what you're asking. Are you referring to training in medical school? Are you referring to the early years of one's practice as a gastroenterologist? What exactly are you referring to?
I think it's both the research and practice. So some diseases are probably the same disease, just coming up in different organs. Like, for example, Prof said EGFR, and the EGFR breast cancer and the EGFR lung cancer are more similar to each other than the 2 different types of breast cancer. And sometimes I feel we are looking too much at the phenotype of the disease rather than the underlying cause.
Well, I mean, I think if you can, you certainly should be looking at the underlying cause of disease. I mean, that's essential to know the pathophysiology. By understanding the underlying cause of a disease, I mean, that's the only way you're going to be able to develop the tools to treat that disease effectively, whether it's by surgical techniques or whether it's, you know, through the use of drugs and so on and so forth. So, I mean, I'm a pharmacologist, so, you know, for me, it's all about mechanism. How this works, why does it work, mechanism of action, of drugs, of chemicals, or the mechanism by which a disease develops. So the way I look at it, mechanisms are very important.
I think we've gone over time already. I wouldn't.
Not yet.
Not yet.
Not
Oh,
yet.
we haven't.
We have
Amazing.
15 minutes.
15 minutes.
Oh, never mind. First of all, thank you. Thank you for a very interesting panel. I come from Sweden, where we recently had the government funded HPV vaccines for free for a specific age group of women to take to be able to eliminate HPV-induced cervical cancer. And now they've had to extend this period because people weren't taking the vaccines. And when I think of the future of medical research, I think of eliminating disease. So I'd be happy to hear some of your insights about how to bring people closer to these solutions, because especially for vaccines, people are skeptical and they don't really trust the government all of the time. And how we can get to a place where people might trust, quotation mark, medical professionals more, but also still keeping the concept of having the right to choose what to do or not to do. And vaccines have needles, or maybe you don't feel like doing something like that, I hope.
Well, you're asking very, very complicated question. Well, I can tell you that if you're really interested in vaccinations and the fact that you should take vaccines, vaccines are very helpful medically. If you really believe that, don't come to the United States in the next 4 years. You're much better off staying in Sweden, for sure. You know, how we go about that ethically, again, you know, I'm a laboratory researcher, and although I'm a member of the American Philosophical Society, I don't know how to philosophize, sorry, about any of that. But it's really important. It's such a shame that so many people, so many governments don't understand the value of vaccines. I mean, that's an incredible tool we have to prevent, to treat, to eliminate so many different kinds of diseases. And I just sympathize, you know, with those of you who sometimes have difficulty getting these vaccines. I mean, that's really all I can say about that. But looks like Aaron is ready to say something.
Well, it's not only President Trump. I think that Sweden, the head of the public health in Sweden, made a grave mistake during COVID-19, which also shook the trust of the public in the system. And Sweden had many more dead on the average per capita than certainly Israel, that took it very seriously. But so, you know, but he didn't do it maliciously. He thought that this will be the right policy, and it turned out to be wrong. But I think that, as Lou said, you know, vaccine is the magic drug. I mean, all over. Think about it. The fact that you are all sitting here, well, 3 years ago we couldn't sit here because of COVID, is due to vaccine, right? We were sitting at home, zooming at one another, looking at screens of computers that looks like stamps, that you see a face protruding out of each stamp and talking from remote. And it's vaccine. And not only that, but the vaccine against COVID that was developed by Moderna and by Pfizer really made a major breakthrough. We are still to see the result of it. Because until then, all the vaccines, think about polio, you need viruses and attenuated viruses, and remember polio, remember the Salk vaccine and the Sabin vaccine, and remember all the other vaccines that needed eggs and purification and so on. And here, at the strike of a keyboard, you get a vaccine. You need 4 bottles with 4 bases, and you strike A, T, C, G, 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 A, C, G, G, and you get a vaccine. And you know, Moderna worked on it for many years, but they didn't dare to go to the market. But all of a sudden, COVID came and they said, we need to hurry because, you know, the epidemic is, disseminated in a vast speed, in a huge speed, and it was successful. It still has to be seen that it will be useful also in cancer and so on, but there are good signs. So I think that, you know, this is a lesson to be learned. And as for vaccination hesitancy, that struck all countries, but to a different extent. The United States suffered more than other countries. It depends. It's education. It's really education. We need to educate the people that polio is out and cholera. You know, it's coming back here and there in underdeveloped countries, but overall, you know, think of the subways in all the world that are super loaded with people during rush hours. Think of theater, think of streets, think of, I mean, we are free society because of vaccines, just because of vaccines. I think that this is one of the major achievements of science and of public health. And we need to educate the people that all the rumors, of course, the social networks play the wrong role here. You know, disseminating, you know, it's affect fertility and it affect that, and you'll get, yeah, there are complications, but there are complications of flying because airplanes crash, and there are complications of driving cars because they make accidents and they pollute the air, and there are many complications to, you know, to the benefits and to the amenities of life, but we live with it, and vaccines are really a magic tool that enable us to be here together, to live longer, to avoid preventive. You know, we are talking preventive medicine. This is the ultimate tool to prevent diseases. So it's education at the end of the day.
I would just add one word. We should shut down social media.
I agree.
Yes, yes.
Thank you.
Thank you again for your time. At a more laboratory level question, there are clear trends in biomedical research where methodologies such as RNA sequencing, omics, CRISPR gain and lose popularity as the costs of doing these experiments fall and new technologies arise. So my question is, what tools or technologies have you seen in the current research landscape that seems the most promising to you that researchers like us can use to help us answer new novel questions or address medical research and healthcare?
Let me answer you in one sentence. In order to use a tool, you need to have a question. And the tool is only to solve the question. So you have to find a good question. The tool is just a tool.
Okay, I would say you should certainly explore AlphaFold 3, because once you can predict structures for proteins and you can predict how they will interact with other compounds, you have a tremendous tool that you can use. It really is incredible, and AlphaFold 3 is much better than the previous versions of AlphaFold. At the moment, it's still a little bit restricted because they don't want the pharmaceutical companies to benefit from it without taking out licenses and so on. But it's going to make a huge difference to our ability to understand how enzymes work, how proteins work, and also to develop new ones and to get better drugs.
Thank you.
Hi, thanks for the great panel discussion. Juliette Fouché from the Karolinska Institute. I wanted to bounce back on a comment from Dr. Ciechanover. So I'm working with ALS patients, and in the community we see a lot of clinicians offering pushback against genetic testing because we don't have a cure for ALS. And in parallel, we have the arrival of precision medicine and precision medicine clinical trials that require a specific genetic mutation to be eligible for the trial. So right now we are in a situation where those patients depend uniquely on pharmaceutical companies to receive genetic testing, without any genetic counselor to then welcome the result and the potential psychological impact of the result. So what's your take, specifically ethically, when it comes to the role of pharma companies in genetic testing?
I think that Rich commented on it already, that the pharma company are a commercial company, and again, I want everybody to ask the question, even if my friends agree, to slide a little bit beyond time, because your part is much more important than ours. The government in any country, in any democracy, therefore I emphasize the role of democracy in ethics and so on, is responsible for our education, for our health. They build hospitals, they have public health care systems, and education. So education, security, military, and infrastructure and so on. One thing they are not responsible, drug development. So the companies are developing drug according to their interest. And Rich mentioned antibiotics. Why the companies should develop antibiotics if you develop resistance, and if you have an infection and you use the antibiotic for 5 days and it's gone? They want you to use an antihypertensive drug for the rest of your life. That every month you'll go to the pharmacy and buy a $100 antihypertensive drug and live for 5 decades with it. The same for anti-high cholesterol and so on and so forth. So maybe, as Rich probably think the same or similarly, the government should be also responsible for drug development, either by incentivation of a pharma company or by making them public or encourage public companies or subsidizing or doing something. For example, some drugs cost millions of dollars. Think about the drug that Rich mentioned on SMA, on spinal muscular atrophy. I mean, no country can afford it, no insurance company can afford it. And also drugs that are not being developed by companies, like antibiotics. So it's going both ways. Either very expensive drug on one hand, or drugs that are not being developed but are at a great need. So maybe it's time for the governments to walk in.
Thank you.
Okay, let me say one thing about ALS. I actually consult with a company in Aaron too called Elysium Health. They have a product called GYSS, and there have been some clinical trials done previously, and now there is one big one underway in Norway showing that GYSS can actually hold back the symptoms of ALS. There was a very convincing study done in Spain, so you know about that already, yes?
Yeah.
Okay.
I'm defending on the topic next month, so.
Thank you so much for your insight. My question is on the sharing of patient samples and materials and reagents. So when you look at the historical papers, you find that labs from Asia, from Europe, from America were transferring samples from one center to another center. However, in the recent decades, because of issues of ethics, patient consent, and commercialization concerns, this is getting increasingly difficult. So I wanted to ask your thoughts on this.
You're just talking about sharing materials?
Yes.
Yeah, you should do it. Period. I mean, this is how progress is made. You know, when we first discovered restriction enzymes, we gave them to anybody who wanted them. We gave the strains. We told people how to make their own if they wanted to. You should do it. It's a good thing. Cooperation and sharing is much better than competition.
With, of course, a small limitation, and that, intellectual properties. And so in the scientific community, you should share anything. I mean, there are some periodicals like Cell publishing companies that forces you to share. If you publish something, you sign that everybody that asks you for this reagent, you should share it. Of course, if you raise some rare antibody and the quantity is limited, it limits you. But if you have a cDNA, a plasmid, you can, no problem. Once it comes to intellectual property that researchers and institutes are trying to protect, then it's another issue. You should share it, but for academic use. Not, people cannot take your plasmid and make a drug out of it and sell it for billions of dollars. That doesn't make any sense. So, but within the scientific community, Rich is right, we should share everything, everything.
Thank you very much.
Okay.
So thank you very much, everyone, for your very interesting questions. I think they were tougher ones than my questions. So maybe to wrap up this session, I will get each one of you maybe to share one tip with our young audience, you know, an ingredient for success in a research career.
Well, perhaps I can start off. Most important ingredient for success. Well, I think we've each touched on this already, and that is being curious. I think curiosity is probably the most important thing that I can attribute my success as a researcher to. I mean, being curious, asking the right questions, attempting to answer those questions in a logical, straightforward manner. I think this is very exciting. Once you begin this and you see that you're on the right track, that's going to increase your motivation, and perhaps to the point where you'll have passion to do what you want to do. And you need, most importantly, you need to enjoy what you're doing. If you're just viewing your basic research as a job, you know, something you need to do because if you don't do it, you won't get tenure at your university or whatever. You won't keep your job at a pharmaceutical company. You know, you don't want to go down that particular avenue. You want to be excited about what you're doing, and that's going to be, I think, the key to your success in the future.
I would reiterate that. Just be passionate about what you do. If you're not passionate, find something else to do. If you go home on a Friday evening and you're concerned about how long it's going to take you to get back to work, you found the right job. If you go home on a Friday and say, oh my God, I got to work on Monday, you're in the wrong job. Change it.
I'll repeat what I said to the students that I met yesterday. Choose something that will not be your job or profession, but will be your hobby. And that will keep you running for decades with high excitement, passion, perseverance, and happiness. And I have nothing to wish you but a great luck in your career. You're really lucky to be here today. In this, when I was a grad student, I never left my country. You have now much greater opportunities. There is another opportunity in Lindau, in Germany, which also is a place of meeting of Nobel laureates and many other students coming from 100 countries and so on. So these forums are great, not only for meeting us, but for meeting your colleagues and to network, disseminate, and these connections will keep you for a long time. So all the best to you.
Thank you very much. So with that, we want to thank our three very esteemed panelists. So thank you to them once again, and we shall wrap up the session. Yeah, thank you.